Hi everyone, my name is Elizabeth Winkle, and today I'm going to be talking about animal use in scientific experiments. So, first off, what type of animals are used in scientific experiments? So, when we refer to animals and their use in scientific experiments, we are most often talking about model organisms. And a model organism is essentially any non-human living creature that is used to study specific scientific processes, whether that's at the very small level, like your genetics, or on more physiological, which is body processes. They are used in many different scientific experiments because findings from model organisms are very easily applied to other things like humans. So, some common model organisms that are going to be used in science are things like mice, fruit flies, and roundworms. So, just real quickly, why do we use model organisms instead of something like just bacteria on a plate? Well, the reason why we use them is because they're extremely easy to maintain. They require very little input from the scientists themselves. They have very short breeding cycles, so you're able to study many generations of them in a very short time. They also come at a very low cost for researchers, which allows them to be very accessible to many different labs across the country and then across the world. They also can be used to study diseases or medications, which makes them extremely important for us as humans because a lot of the scientific breakthroughs, whether it's from cancer research to things like Tylenol, a lot of that research has actually come from experiments on model organisms. They also are very easy to replicate when it comes to experiments on them, and that makes it very important because in science, we want all of our results to be easily replicable. So you might be wondering, as model organisms are animals and we never want to mistreat an animal, how do we keep model organisms safe? Well, the guiding principle of any experiment on model organisms, which scientists always have to keep in mind, is going to be the three R's, as well as just overall a sense of ethics and morality. And what the three R's are is replacement, which that means whenever it's possible, a scientist is going to use an alternative model, whether that be modeling the experiment on a computer or running something on a petri dish. The next is going to be reduction, which is just simply reducing the number of animals used. So, for example, instead of using 500 mice, a researcher will attempt to get all of their findings with as few as 15 mice. And then lastly, it is going to be refinement, which that is just simply improving the animal's welfare whenever possible in an experiment. So, just to illustrate really quickly how model organisms are applicable all across our lives, I'm going to be looking at a very local example, which is a Lazier lab at UNC and how they use mice in their experiments. So, what this lab does is they study basically the immune reactions and the mechanisms and how your immune system fights herpes simplex virus, also known as HSV, or just more simply herpes. What they do is they use a very common model organism, which is a mice, which scientifically we refer to as Mus musculus. And what they have done is they found a very important way to model herpes in mice by simply plucking the fur on the back legs of the mice. Now, this is really important because before this work, there have been very few mice models for HSV, but mice are one of our most important models for things like drugs and other physiological, which is full body function, as I mentioned earlier. And what they were able to do is reactivate inactive herpes, and they just found a completely new model. We're then able to apply all of this work to humans and just model how herpes progresses through a human's lifespan, which is super important. And how they apply the three R's is whenever they plucked the mice and infected them, they made sure to use anesthesia, both numbing the mice and putting them under sedation. They also used the minimum number of mice possible, reducing it to around 20. They limited the infection to a small area, as you can see in the picture, just a small part of their back flank or their back legs was used. And then they closely monitored the health of the mice. So as you can see from both my introduction to model organisms and my example in the Lazier lab, using animals in scientific experiments is extremely important. Without their work, we would not be able to have many of the breakthroughs that we have today, whether it's ranging from fields of cancer to even the simple things like painkillers. Model organisms are extremely important, and I hope that you understand why we use them and some of their benefits after today. Thank you.